Hello, everyone. Thank you for your interest in our poster titled Hybridization LC-MS for sensitive quantitation of antisense oligonucleotide in plasma and dry blood microsamples, presented at the 2025 WRIB conference. My name is Ming-Luan Chen. I am a principal research scientist at the Altasciences. It's my pleasure to introduce our study and discuss its novelties with you today. Antisense oligonucleotide ASOs are powerful therapeutics agents that target disease-related genes. However, accurately measuring ASO concentration in biological samples is a challenge due to their low abundance and the complexities of sample matrices. Current analytical methods often lack sensitivity or require large plasma volumes, making routine monitoring difficult, especially in the decentralized clinical trials where convenient sampling is crucial. Our study addresses these challenges by developing optimized hybridization LC-MS methods that enables high sensitivity quantitation of ASOs in both plasma and dry blood microsamples. By integrating sequence-specific hybridization, extraction, and post-extraction divert valve, we significantly improve assay performance, offering a patient-friendly alternative for clinical studies. To quantify ASO and their primary N-1 metabolites, we apply a systematic workflow, which is shown in Figure 1. First, we connected the plasma sample using 100 microliters volume, or we using 10 microliters of blood to make the dry blood microsamples with Container B device. Then we performed selective extraction using the biotinylated capture probe designed for hybridization-based extraction, which ensures high selectivity by removing the on-target oligonucleotides. Finally, we analyzed these samples using LC-MS with the iron pairing chromatography. As shown in Table 1, we optimized the MRM parameters to achieve high sensitivity and the specificity for ASO quantitation. A key improvement in our approach was the introduction of the post-injection divert valve as illustrated in Figure 2, which significantly enhanced chromatographic performance. This improvement boosted peak sharpness and the resolution, resulting in a better sensitivity and reproducibility. Figure 3 highlights the improvements in sensitivity and the peak sharpness, particularly at a low flow rate, where the conventional method often suffer from poor resolution and the single suppressions. Our method demonstrated exceptional performance in the ASO bioanalysis. The lower limit of quantitation at LOQ was 50 picogram per ml for plasma and 0.5 nanogram per ml for the dry blood microsamples, as shown in Figure 4 and 5. The precision and accuracy result for dry blood microsamples summarized in Table 2, where we see acceptance criteria ranging from 80% to 120%. The method was also evaluated for hemocratic bias in Table 3 and the matrix effect in Table 5, with no significant deviation observed across different hemocratic levels and the blood donors, confirming the robustness for clinical applications. The precision and accuracy result for plasma samples summarized in Table 5, where we see acceptance criteria from 80% to 120%. The method was also evaluated for matrix effect from 10 different lots of plasma donors, including lipomic and hemolyzed lot. As illustrated in Table 6, no significant deviations were observed across these lots, confirming the robustness of this analytical methods. This method quantification demonstrated the reliable quantitation during the clinical sample analysis. In summary, our hybridization LC-MS-MS method represents a significant advancement in ASO bioanalysis by offering unmatched sensitivity, minimal sample requirements, and enhanced analytical performance. We provide practical and reliable tools for ASO, PK, and metabolism studies. The integration of the dry blood microsamples and the hybridization-based enrichment makes the method highly suitable for both centralized and decentralized clinical trials. Thank you for your attention. I will be happy to answer any questions or discuss potential application of this method with you. Please feel free to reach out. Thank you very much.